So welcome back to Orthodoxy 201. We are in the chapter The Logos. Of course, Jesus Christ is the Logos, the Word of God, the one who created the world and brought forth life. And we have been looking at scientific discoveries in the last 100 or so years that point to God as the creator, as the one who brought forth life, brought forth things from non-existence into being, created the universe in a very finely tuned way, and then established on our planet life in its great variety and development and beauty. And we've been discussing different approaches to how to understand the world. One, of course, is a Christian viewpoint, and we have looked historically at the early scientists who shared that perspective. They believed that God did create the world to be intelligible, and that they thought that by understanding the world and God's creation, they could understand the creator, they could understand the Logos, they could come to know Christ. And that theory prevailed and for some time, but then was replaced by naturalistic, materialistic theories that were atheistic. They excluded God, and they had to answer then the questions of how the universe exists and how there's life in such great variety and there's such order and harmony and beauty in the world. So we've been discussing in the last few sessions these big questions. And last time we looked at the origin of life and chronologically subsequent theory, the theory of evolution. So the theory of abiogenesis is one where we get life from non-life. And it largely, the scientists of the materialistic bent believe in some kind of primordial soup, and life emerged out of this soup. And then with the theory of evolution, developed from simple to complex organisms. We'll be discussing the actual science behind those two theories. We did a little last time, we'll do some this time, and then continue probably for 2 more sessions on that. And then we'll kind of wrap up on the scientific presentation. But we looked at this idea of the primordial soup in an experiment that was done in 1953, which was done in the lab, and they noticed some chemical reactions and thought that they had all of the building blocks and the process by which they could bring forth life. That experiment was rather underwhelming as far as the ability to convince anyone that the great complexity that we see in the cell could come forth from a primordial soup. We discussed the history of the understanding of the cell being some kind of simple blob to a great machine, a machine that is so small that you can't really see it without a microscope, yet so complex that it rivals what you might see in the greatest cities of the day. And we look at different parts of the cell here in this diagram. And then we watched a video, which was wonderful short film, about 10 minutes long, on the cell. And it introduced us in, I think, a very visual, illustrative way to the great wonder, really, of the cell and its great complexity. We spoke a little bit about Dr. James Tour, who is a leading synthetic organic chemist and nanotechnologist. He is well published. He has numerous patents, both that he's already received and those that are in progress. And he, being an expert on the study of abiogenesis, has really called out his scientific community to say, put up or shut up. Because a lot of people have been claiming that in the lab they can bring forth life, and yet he contends and really proves that that's the furthest thing from the truth. That many times when people speak, they're speaking more from an ideology or maybe their hopes rather than observational science and things that can actually be proven. And this, he contends, is definitely the case with abiogenesis, that we are nowhere near close to the first step in creating life. He even proposes the idea that if you were to get all of the scientists, the best scientists of the world together and build a laboratory sparing no expense, you have all of the material resources available at your disposal, and give them the sole function, the sole purpose of producing life in the lab, that they couldn't do it. They couldn't even get the building blocks working. And that would take a whole lot of resources and a whole lot of intelligence. And mustering all of that, they couldn't get anywhere close to bringing forth life. And he's contending not in any kind of complex form, but the simplest of forms, right? So it's highly improbable. And we've been looking at probability as one of the key aspects to discerning what is true from what is false. He says it's so astronomically improbable that you would at some point even get life in a laboratory, that it makes it infinitely improbable that you would bring forth life in an unintended manner by undirected natural processes. And being a chemist, he points out that it's not a problem that'll be solved by having enough time. If you just wait around and eventually chemicals will bump into each other and they'll start to make connections and life will form. It doesn't work that way because chemicals degrade. And scientists in the lab work very hard to stabilize their chemicals so that they don't degrade. So, and these chemicals, some degrade in a matter of minutes or hours. So adding millions or billions or trillions of years to the equation actually doesn't help anything. It would probably make things much more difficult because time is not the answer to the problem. Time is part of the problem when you're dealing with chemistry. Okay, and then we looked at DNA, which the cell has such great complexity, but that complexity of the cell is also tied to the complexity of DNA. DNA is the instruction manual for all living beings, all living things. And it's basically all of the information, the code behind life. It instructs how life is to be created, how it's to be sustained. And the code is something that is extremely complex. So Bill Gates has this quote, DNA is like a computer program, but far, far more advanced than any software ever created, right? And almost really any software ever even imagined. Because the kind of complexity and creativity that you see in DNA it's beyond the human level, right? Nobody could try to code in the manner in which DNA is coded. We just don't think that way. We don't have the brains to process at that level. I gave as an example, if you were to read a book in a library, from page 1 to the last page, it would read a story. It would give you some information. But if you were to take that same book and you were to jump around and pull out the 27th word on different pages, that would also lead you to read another story. It would give you another novel. And if you were to pull out books from the library and go to page 167, word 27, that would also give you another story. So the DNA is not just coded in a linear fashion. It's coded at multiple levels in multiple ways. And it all connects and fits together like a puzzle. And a puzzle that really we're not equipped. We don't have the intellectual capacity or the CPU power to even understand fully ourselves, let alone create. Right? So the intelligence behind the information is superhuman. And we're in the AI kind of era. AI is not able to do the stuff that we see in DNA. All right, and then we left off here and we're going to talk a little bit about single-celled organisms. And this is both to consider the question if single-celled organisms could arise from a primordial soup, right? Because we're talking about the simplest form of life. It could arise from primordial soup. This kind of pool of liquid that's somehow struck by lightning and brings forth a single-celled organism. We'll see if that's possible, if that's rational and reasonable. And then we'll talk a little bit about the theory that single-celled organisms then, through the process of evolution, developed into multi-celled organisms and the great complexity and variety that we see today. So can I ask, Suzanne, can you start us off, please?
Single-cell organisms. The amount of DNA in a single-celled organism can vary widely depending on the type of organism. Here are a few examples. Bacteria. Most bacteria have a single circular chromosome. For instance, the well-studied bacterium E. coli has about 4.6 million base pairs of DNA. Yeast. A single-celled fungus used in baking and brewing has about 12 million base pairs of DNA across 16 linear chromosomes. Amoeba. Some single-celled eukaryotes have much more DNA. For example, the amoeba dubia has one of the largest known genomes with around 670 billion base pairs of DNA. The amount of DNA in single-celled organisms varies greatly, ranging from a few million base pairs in bacteria to hundreds of billions in some amoebas.
Thank you, Suzanne. So even these single-celled organisms are just jam-packed with information, right? And they're so small, they're so tiny, and yet they're holding so much information. I have a number of different hard drives, and I can't hold on my large hard drives the amount of information that is held in such a small little cell, right? It's unbelievable, the design at the nano level of the cell and DNA. So I wanted to show a short video. This is shorter than last time, just a few minutes, and it'll give us an introduction to DNA. So let's watch that.
Welcome to this informative video on the structure and function of DNA. We are going to learn this topic in 2 sessions. In the first video, we will dive deep into the structure of deoxyribonucleic acid to explore its intricate structure and its significance in the realm of genetics and molecular biology, whereas in second video, we will learn about its function. The discovery of DNA is credited to several key scientists, including Friedrich Miescher, who isolated nuclein from white blood cells in 1869. However, the breakthrough came in 1953 when James Watson and Francis Crick proposed the double helix structure of DNA. Their work built upon Rosalind Franklin's X-ray diffraction data, which revealed the helical nature of DNA. DNA is composed of 4 nucleotide bases: adenine, cytosine, guanine, and thymine. These nucleotides are connected to a sugar phosphate backbone, forming a single strand of DNA. The specific sequence of these bases holds the genetic information necessary for an organism's development and functioning. The iconic double helix structure of DNA consists of 2 complementary strands that coil around each other in a twisted ladder-like formation. The bases on each strand interact through hydrogen bonding. Adenine pairs with thymine and cytosine pairs with guanine. This complementary base pairing provides DNA with remarkable stability and facilitates DNA replication and protein synthesis. The double helix structure creates grooves along the DNA molecule. The larger groove is called the major groove, while the smaller one is known as the minor groove. These grooves play crucial roles in various DNA-protein interactions, such as transcription factors binding to specific DNA sequences and initiating gene expression. DNA can undergo supercoiling, a process in which the helix twists upon itself to form a more compact structure. Supercoiling is essential for the packaging of DNA inside cells and plays a pivotal role in regulating gene expression and replication. In eukaryotic cells, DNA is organized into chromatin, a complex of DNA and proteins. Nucleosomes are the fundamental units of chromatin, consisting of DNA wrapped around histone proteins. This structure helps regulate gene expression and provides stability to the DNA while allowing accessibility for essential cellular processes. As cells divide and prepare for cell division, the long DNA molecules must be compacted further to fit within the cell's nucleus. This process involves organizing the chromatin into chromosomes. During cell division, the chromatin condenses into visible, condensed structures known as chromosomes. Each chromosome is made up of two sister chromatids, which are identical copies of the DNA. At the ends of chromosomes, there are specialized regions called telomeres. Telomeres serve to protect the vital genetic information by preventing the loss of DNA during replication. They also play a role in cellular aging and have implications in diseases such as cancer. The centromere, on the other hand, is a region on the chromosome that ensures proper segregation during cell division. It serves as an attachment point for the spindle fibers that help pull apart the sister chromatids during mitosis or meiosis. While the fundamental structure of DNA is conserved across all life forms, there are variations in the DNA sequences and the organization of genetic material. Prokaryotic organisms, such as bacteria, have a simpler organization with a single circular chromosome, while eukaryotic organisms, including plants, animals, and fungi, have linear chromosomes contained within a nucleus. DNA is continuously exposed to damaging agents, both internal and external. To maintain the integrity of the genetic code, cells possess an array of DNA repair mechanisms, including base excision repair, nucleotide excision repair, and mismatch repair. These repair pathways are crucial for preventing mutations and maintaining overall genome stability. Understanding the structure of DNA is paramount to grasp the fundamental principles of genetics and molecular biology. The double helix structure, along with its various interactions, is a cornerstone of life on Earth.
Okay, so that video hopefully was interesting for people. It did say that there was going to be a second part. We won't watch the second part. I think, though, that what we saw just now hopefully is enough to get across the point that it's not just like a little bit of information that's present in DNA. Like you roll dice and you throw it in the corner and it's going to be like a 1 through a 6. This is a vast amount of information that has creation and retention and error correcting and all of these processes that are happening at the chemical molecular level. And it's all part of the cell, right? And this whole system is integrated in a way that lightning hitting the primordial soup probably couldn't accomplish if you're looking at it from kind of a rational perspective. But I do want to talk a little bit about the origins of DNA, and I have some presented here. But first, let me note for the readers that video was helpful because it also did give the proper pronunciation on some of these terms. So hopefully the readers are paying attention during that short video. Just teasing. Okay, so can we go to Georgianne for this next one, please?
Okay. Origins of DNA. The origin of DNA is a topic of significant scientific inquiry and debate. And while the origin of DNA is unknown, the following hypotheses exist. Primordial soup hypothesis. Life began in a mixture of organic molecules formed from simple compounds in the early Earth's atmosphere, leading to nucleic acids. RNA world hypothesis. RNA was likely the first genetic material, with DNA evolving later for greater stability and efficiency. Clay hypothesis. Complex organic molecules formed on clay mineral surfaces, facilitating their organization. Hydrothermal vent hypothesis. Life may have originated at hydrothermal vents, where mineral-rich superheated water provided favorable conditions. Extraterrestrial origin. Building blocks of DNA could have arrived on Earth via comets or meteorites. Intelligent design. As minds create, organize, and interpret information, intelligent design posits that DNA's complexity and information content indicate an intelligent cause, not random natural processes or evolution.
Thank you, Georgianne. So these are the main theories. Obviously, the top 5 that are listed there are naturalistic theories, right? And the bottom one, intelligent design, is the traditional theory from the early scientific revolution, with those believing Christians who pioneered science, and also is making a return today. I've spoken about in the past, and we'll introduce maybe next time, a text by Dr. Stephen Meyer on the return of the God hypothesis. It's a wonderful book dealing with intelligent design, summarizing some of the things that we're talking about and also getting into much, much greater detail. So intelligent design has been a theory that existed really at the beginning of the scientific revolution and is making a comeback now, especially as we see how much information is there is coded in the very fabric of life. And information, whenever we see it, it comes from a source. Anything that is intelligible comes from a mind, right? So some examples here. So you see a piece of music that would come from a composer, computer code from somebody who's a software developer, blueprints, the architect, even this kind of like alphabet soup type thing, or SpaghettiOs or whatever that is. That message was written by a mind. You're not gonna, if you find that in your soup in the morning and you're wondering like, how did that get there? Somebody probably set that up for you, right? And then of course you see Mount Rushmore, which presumably wasn't formed by just wind and natural erosion, right? Somebody carved in, and actually that is not Mount Rushmore. I think that's called Mount Trumpmore. I found that online, adding a little humor to this. But the idea that you find a rock formation like that without somebody doing that on purpose is preposterous. Much more so, and perhaps outdoing all of the other examples here, when you look at DNA. DNA is not going to happen at random. It's not going to be an accident. There is so much information, and again, put there by not just an intellect, but really a super intellect. So all of those other examples, it's common sense, much more so in the case of DNA. Okay, but let's actually look at the theory of evolution, and we'll talk about genetic mutations, talk about natural selection, and we'll talk about time, because these are really the 3 main components to the theory of evolution. So Marion, can you take genetic mutations for us, please?
Yes. Okay. Genetic mutations. While it is sometimes said that mutations create new genetic information, they do so only by modifying or repurposing existing information rather than creating entirely new sequences from scratch. Gene mutations degrade DNA by causing point mutations, substitutions, insertions, deletions, and large-scale changes like duplications or translocations. These alterations disrupt gene function, lead to faulty proteins, and can impair DNA repair mechanisms, resulting in structural damage and loss of genetic information over time. When these mutations accumulate, this leads to progressive degradation of the genome, loss of genetic information, and the potential development of various diseases, including cancer, genetic disorders, and age-related conditions. Additionally, DNA mutations alone would not be enough to generate new anatomical improvements. Successful anatomical changes would also require precise gene regulation, proper protein function, cell differentiation, tissue organization, and effective cellular communication, all of which must work together to produce beneficial traits. This would be highly improbably, as DNA mutations are random and not inherently coordinated across different organs.
Thank you, Marion. So when we're talking about a mutation, we're talking about a change that would take place in the DNA code, which would happen at random. It wouldn't be a purposeful change, like a scientist might try to modify the genetic code in a lab. This would be something that just happens at random. And when you change a piece of music, you change text in a novel, you change code in software. When you change things, they actually, at random, they actually don't improve the music, the novel, or the code. It actually degrades it. It lessens it. It destroys it. And if by chance you were to, at random, make a change that potentially could make an improvement, and that's possible, right? We have to admit that that's possible. Like if you wanted to, at random, just take one word out of a novel and put in another word, there is a chance, although small, that you would find a better word to plug in, right? Overwhelmingly, it would be the case that you did not, but presumably it's possible that you could. That still wouldn't be enough to actually bring about a change, because we're not just talking about things in theory. Life has a concrete, tangible aspect to it. It's like this: if you're at a job site and you look at the blueprints for, I don't know, a home or something like that, and you decide, I'm going to simulate a positive genetic mutation with this blueprint, and I'm going to draw on it another wing, and I'm going to put in this an indoor pool, and that's going to improve the structure and the home. Well, that's all great, except for where are you going to get the materials, right? Because it's not enough just to change the blueprints. If something is actually going to form, you're going to have the materials for that. So this isn't just a question of information. It's information that is synchronized to the biology, right? To the anatomy of whatever life is being formed or maintained. So when we're looking at genetic mutations, though, that's one of the key components with regard to the theory of evolution. And we'll talk a little bit more about them. But we have to also discuss natural selection. So can I ask to go back to Suzanne for this one, please?
Natural selection, although rare, in some cases, mutations can lead to the development of new traits or adaptations that were not present before. For example, mutations in bacteria can confer antibiotic resistance, which represents a new capability not previously present in the population. However, this is a result of breaking functionality, not creating new complex features. For example, chloroquine resistance in the malaria parasite Plasmodium is due to a fault in a transportation protein that moves the poison into the organism's vacuole used to store waste. Dr. Michael Bay likens such improvements to the struggle of trench warfare, where defending forces will destroy their own bridge or blow up a road to impede the enemy's advance. It is not really an arms race, because in an arms race, the opposing forces invent new weapons. The natural processes, evolution, operating in Plasmodium, have not invented new weapons. Dr. Robert Carter. Mutations, even when acted upon by natural selection and time, steadily degrade a genome, increasing genetic entropy, and therefore cannot produce a better organism in the long run. We observe, therefore, the exact opposite of evolution, devolution.
Thank you, Suzanne. All right, so sometimes we hear about like a positive mutation or something that is beneficial, and that can be the case. And there's an example that's given here where a change takes place and it changes the functioning of that organism in a way that is beneficial. But it's actually not improving the organism in and of itself. It's simply changing it so that it functions in a different way with its environment. It's not creating more information. It's not adding more function. It's degrading. And that degradation can serve a positive purpose. So we do see examples, and a number of different examples, of how the degradation of an organism allows it to survive. We do see that, and that acted upon by natural selection does change the future of that species. But it's not changing the trajectory in the sense of moving to greater complexity or a greater functionality, or what we would call the evolution of the species. It would actually go in the opposite way, degrading it and leading to devolution. And that's just the nature of changing things at random. And I have this little meme here with Bill Gates, not to poke fun at Windows or anything like that, but there's the expression, it's not a bug, it's a feature, right? That's what we're talking about here, where something actually doesn't work anymore, but it's somehow to the advantage of the organism with regard to its environment. And there are examples of that. Overwhelmingly, though, there are more examples where an organism suffers from genetic mutations. And that's why in the biology, in the way that DNA perpetuates itself, there are maintenance and error correcting systems that are constantly at play. And they, by the way, prioritize those genetic instructions which are more crucial to the survival of the organism. So things that would be, for instance, creating the main systems, whether it be vascular or cardio or whatever the case is for the organism, those parts of the DNA are going to be given greater priority. Where things like eye color or other maybe not so crucial components to the organism don't get that same level of error checking. So natural selection, though, and genetic mutations do oversee the downgrade of an organism. And not just an organism, all organisms. And there's a reason for that. Because the second law of thermodynamics is the law of entropy. And that is the law of degradation. In theology, we call it the law of corruption, right? Which resulted from sin. Sin is separation from God. God is the logos, right? He's the great designer. He's the one who imprints his plan and his blueprint in the very fabric of life. And that's done through the logi, right? The words, so to speak, the code that we see so beautifully in DNA. All of that is imprinted by God. But when man turns away from God, he turns away from the logos, and he falls away from his purpose, his meaning, his very constitution. The very words, the logi within man, cease to function as they should. They become corrupted, and he begins to degrade. He begins to die. And that was what was told of Adam and Eve when they would come to know the knowledge of good and evil by, in disobedience, turning on the logos, right? That the day that they would turn on the word of God, they wouldn't listen to the word of God, they would surely die. And immediately there is this law of corruption that enters into the world. And that's an observable law where the theory of evolution actually is not observable. It's theoretical science, but a bad theory. And when the two butt heads, the law of entropy wins, right? So let's talk a little bit more about that. Who's up next? Is it Georgianne?
Yes. Okay. Survival, but not arrival. Genetic mutations and natural selection do not create new information nor new anatomical structures. Genetic mutations show how living creatures devolve over time, and natural selection shows which organisms survive the test of time. However, genetic mutations are not creative, but destructive. And as Dutch botanist and genetics pioneer Hugo de Vries famously said, natural selection may explain the survival of the fittest, but it cannot explain the arrival of the fittest. The law of entropy, the second law of thermodynamics, asserts that orderly things over time become disorderly. Cars wear out, flowers die, and people get old. This leads some to predict that the universe will end in heat death because, according to the second law of thermodynamics, entropy and disorder will continue to increase as time passes. The theory of evolution suggests that life becomes more ordered as time goes on. However, this is not observed in biology nor any other scientific discipline, and this hypothesis violates the law of entropy.
Thank you, Georgianne. So let me pose a question and offer a response to that. So how is it, though, that as time passes, we live longer and we advance in technology, right? And we have all of these developments. How is it that this is taking place? Well, what is behind that is the passing along of intelligence and from one generation to the next generation. So intelligence can increase. One can become more logical, rational. One can become closer to God, so to speak. And we do see that. So the reason why we live longer is not necessarily because our genetic code is in better shape. That is degrading. What is different is we have better sanitation and we have better medicine, better technology. And for that reason, we're living longer. Are we necessarily more intelligent than our ancestors as far as our genetic capabilities? You can't make that argument looking at DNA. You also can't really make that argument looking at simple, mundane things of life. I'm thinking about, for instance, this is just an illustration. When I was a teenager, I could remember all of my friends' phone numbers. I could just recall them whenever I needed to. I have trouble. I don't even know my kids' phone numbers. I really do not know my kids' phone numbers. And that's the case for many things. We rely on computers, we rely on technology, we rely on all of the things that we have built up over the centuries. And in a sense, we rely on them too much. And we suffer with regard to our own personal development. But if you were to follow this backwards in reverse time, you would see actually that the human being and the genome was in a better place in years past. I mean, you might even say that we would expect to see, if you went back in time, that people lived longer, right? Has anyone heard of any text that talks about people who in the early days lived longer than they do now? I know of a text, the Bible, right? The scriptures talk about that. And we could talk a lot more about that. Right now, we're really just looking at scientific, not just data, but ideologies and things like that. We'll talk more about the scriptures in later classes. But we do have a witness to the fact that entropy does exist, and we were in a better state in the past. And that witness is scriptural, but it's also scientific, right? All right, so let's talk about this idea of irreducible complexity. This is a very important concept that is often attributed to Michael Behe, who has done very fine work on the subject. But it's something that just makes kind of common sense. And that's, by the way, one of the ways that you can tell if something is true, that it makes sense. It's rational, it's logical. And that should be the underpinnings of all science. It should be based in the logos. So he makes the case, though, that these systems that we see, for instance, in the cell or in the way DNA works, that these systems require many parts. And these parts have to be co-working. So much so that if one part or two parts or three parts were missing, you wouldn't have the system. You wouldn't be able to function or to build anything. So let's look at his example of the mousetrap. So Marion, can you take this one for us, please?
Irreducible complexity. This concept, introduced by biochemist Michael Behe, suggests that certain biological systems, e.g. the bacterial flagellum, are too complex to have evolved through natural selection alone because they require all parts to be present simultaneously to function. Behe argues that such systems could not have evolved through successive slight modifications, as proposed by Darwinian evolution, because intermediate stages would be non-functional. To illustrate irreducible complexity, Behe uses the example of a mousetrap with a base, hammer, spring, catch, and trigger. Functional interdependence. All parts of the mousetrap must be present and correctly assembled for it to work. If any single part is missing or not functioning properly, the trap will fail to catch mice. Non-functional intermediate stages. A mousetrap cannot be built by adding parts one by one because intermediate stages without all parts would be non-functional. For example, a base with only a spring and no hammer or catch would not catch mice.
Thank you, Marion. So genetic mutations, even if they, in an improbable manner, lined up to produce multiple benefits over a period of time to an organism, and they started to build, let's say, a mousetrap. So first, you might have the base. Then the next generation, if things really worked out well, now you have a hammer. And the next generation, you have a spring, and then the catch, and then the trigger, right? That actually wouldn't confer any advantage on that organism until all of those parts are there. So natural selection is not going to figure into this because it's actually not going to help the organism until all of the parts have been built. In fact, though, if an organism had a base, a hammer, a spring, and a catch, missing the trigger, all of that would be appendages that would actually harm the functioning of that organism because they would not be doing anything, right? So if you had, I don't know, an extra appendage coming out of the middle of your back that you couldn't control and it couldn't do anything, it had no function, that would actually be dead weight, right? That's how natural selection would operate if you were trying to build these things one at a time. And by the way, the idea of getting all of these successive beneficial mutations still doesn't address the question of, well, where do you get the anatomical materials to build these things, right? So this is just another example, though, of why the theory of evolution simply doesn't work. And let's look at something else with regard to irreducible complexity for a moment. Some of you, probably all of you, have seen at one point or another that film, The Wizard of Oz, right? Where you have Dorothy who just wants to get home, right? But then she comes across the Tin Man, the Lion, the Scarecrow, right? And they're all missing something. There's some key component to them that they don't have, without which presumably they couldn't live, right? So the Lion needed courage, the Tin Man, the heart, the Scarecrow, a mind, right? There were things that they needed. Think about the absence of those crucial elements in any organism. You couldn't imagine it living, right? And there's just series of examples that can be presented when we look at not just the complexity of a complete organism like a human being. When you look at a simple cell or a part of a cell, right? Because a cell has so much complexity that it itself can't exist without key parts, right? It needs all of the pieces of the machinery to function, and it needs all of that code to tell it what to do, right? So all of that is necessary on day one. And that means you must have had a really great lightning strike on that little pond of scum or whatever you got, right? We're really at the heights of irrationality when we look at abiogenesis and we look at the theory of evolution. The science is really strong on this. And yet, by the way, not to keep going back to how the science is misrepresented, but most people don't know anything about this. They're not given this basic information that is irrefutable, right? And I can't help but see how ideology dominates this portion of the scientific world. And it's not uncommon. Ideology dominates so many things in life today. But let's give the theory of evolution its fair due. Let's talk about the one missing element, right? Because we talked about genetic mutation, natural selection. Let's talk a little bit about time. Time will save the day, right? It can happen if there's enough time. So who's up next?
But with enough time. With time, all things are possible. Author unknown. Validity of the statement false. It is purported by evolutionists that given enough time, a single-cell organism, through genetic mutations and natural selection, can in fact evolve into a human being. However, how much time is needed? How much information do mutations have to add to change a minimal microbe into a human? The essential microbe's genome is about 500,000 nucleotides, letters. A human has some 3 billion. So to change the former into the latter, mutations have to invent nearly 3 billion nucleotides, or letters, of information specifications. In terms of proteins alone, a basic microbe has several hundred, but a human over 100,000. Dr. Robert Carter.
Thank you, Suzanne. So we're talking about a vast amount of information, a vast amount of code. And that code, remember, is written in such a manner that it's not just linear, right? But that code specifies things in multiple ways, right? That code simultaneously can be read in multiple ways. And that would have to be generated at random. Okay, so, but if there's enough time, maybe it could happen, right? Maybe if there's enough time. Someone did a calculation on what it would take to try and form one functional protein by chance. Okay, Georgianne?
Okay. Time is actually the problem, not the solution. The longer you wait, the more degradation occurs in the prebiotic chemicals. Time works against the formation of life from non-life. James Tour, synthetic organic chemist and nanotechnologist. Time is often invoked as the hero of the plot for those who espouse materialistic evolution, as if given enough of it, anything is possible. But time is actually the villain, because the more time that passes, the more improbable and unworkable these unguided processes become. Stephen Meyer, PhD, Cambridge. Time is actually the enemy of evolution. As time goes on, genetic entropy builds up. Mutations accumulate faster than selection can remove them. Given enough time, instead of evolution, we get devolution. Dr. John C. Sanford, geneticist. The belief that time is the cure-all for the improbabilities of evolution is like believing that if you roll the dice enough times, you'll eventually get a coherent sentence. Time doesn't fix the problem, it amplifies it. Michael Behe, biochemist.
Thank you, Georgianne. So this biochemist, Douglas Axe, he asked the question, what is the probability of one functional protein forming by chance? The result of that question, the answer to that question, is 1 followed by, I believe that's 27 zeros in years. Okay, now we're running into a little bit of a problem because there's not that many sands in the hourglass, right? Because of the theory of the Big Bang, the universe is estimated to be 13 billion or so years, give or take. Does that get us anywhere close to that number? Not even close, right? And we're dealing with a number that's like incomprehensible just to arrive at one single protein. Cells are made of numerous proteins, right? So mathematically, statistically, probability-wise, right, the theory of evolution is entirely refuted, right? This is as done a deal as imaginable. And yet, in our textbooks in schools, you would think it's just the opposite, right? You would think it's just the opposite. How can that be? I think we have to look beyond the science, and we have to look a little bit more at the way the human mind works, the way societies work, the way the world works, right? And it works in a way that it has turned on God, who is the Logos. It has embraced irrationality, it has embraced falsehood, it has embraced deception. And that is very powerful in this world, so much so that you almost can't see the light, right? People think to be a Christian, it's so hard in today's world because we have no evidence on our side. There's nothing in our favor. It's a dark world out there for the Christian. It's shining so brightly now, like brighter than ever. And yet the cloud that hangs over us, that doesn't allow us to see the light, is this fallen and corrupt world. And the deception that it is being purported at the highest levels. The science speaks so strongly for belief in God that it's overwhelming. Okay, just to nail this point home, and I know we're a little bit over, but I'd like to kind of tie up some loose ends before we go on to the next part of this, because there's even more information to look at. Let's look at the chances again, because maybe something was wrong. Maybe we missed something. So, Marion, I think, are you next?
Yes. Evolution, unfathomably improbable. In the cartoon The Simpsons, Mr. Burns trying to impress Homer with the many absurd features of his vast mansion. Among these is a room containing 1,000 monkeys with 1,000 typewriters, who Burns claims will soon have written the greatest novel known to man. It's a hilariously inexplicable scene, particularly the goofy payoff, "You stupid monkey," when viewers see the overworked simians aren't quite there yet. But what is this gag referencing? It's actually a nod to the infinite monkey theorem, a famous math-based thought experiment. The theory argues that an infinite number of monkeys, given an infinite number of typewriters, would inevitably end up writing every combination of letters, including the collected works of Shakespeare. However, it would likely take all the monkeys in the known universe, and a lot more than the Simpsons 1,000, more than the duration of the universe's existence, to write Hamlet. But there is still a chance that it would happen. Likewise, it is possible that the theory of evolution is true. It is just unfathomably improbable.
Thank you, Marion. So next time you have a conversation with somebody who's laughing at your faith and saying how the science is spoken and it's a done deal, evolution is basically a fact, consider this. Remember the example of the monkeys, right? It is unfathomably improbable that the theory of evolution is actually true, right? You can just say it is false. Like, we can get to the point, by the way, where something is so improbable that you can say that's just it's not happening. It's not possible, right? We can do that, and we should do that. And our scientific community, because we're dealing, right, with science, and we're dealing with factual data, observable data, mathematical equations, probabilities, right? They should be able to produce this for us, right? They should be able to themselves say, the example of the stupid monkeys and the infinite monkey theorem really doesn't even come close to how improbable it is for abiogenesis and for the theory of evolution to be true. They should be able to tell us that, right? But if they can't, you can tell them.